Okay, so I think we'll get started. So I just want to start by saying welcome everyone, and thank you so much for joining us today for the first in a kind of mini two-part Lunch and Learn webinar series on invasive Phragmites. So my name is Elsie Lewison, and I'm the Development Officer at the River Institute. And I think most of you online are probably familiar with us, but if not, we're a not-for-profit environmental research, education, and outreach center based in Cornwall, Ontario. And we're currently involved in a couple of ongoing collaborative projects related to invasive Phragmites. So this includes a project supported by the Canada Water Agency's Eco Action Program. So I want to acknowledge their financial support. So we're also involved in the Eastern Ontario Phragmites Management Area, or PMA, and this is one of a number of PMAs under the broader Ontario Phragmites Action Program. And our PMA encompasses the Raisin region and most of South Nation watersheds. So if you're not yet involved in a PMA and are interested in becoming involved, please feel free to reach out to me. I can put my email in the chat. And if you contact me, I can put you in touch with the right people. Before we get started, I also just wanted to plug a couple of upcoming events. The first one, Megan, if you want to go to the next slide. So the first one is the next installment in our mini-series, which will be taking place from noon until 1 p.m. on Wednesday, February 11th. So for this webinar, we have Victoria Kellenyuk from Niagara Parks, who will be providing an overview of a Phragmites removal project using goat grazing. So that should be really interesting, and I hope you consider joining. I'll put the link to register for that webinar in the chat as well. And then for those of you in or around Cornwall, the River Institute is also hosting a Science and Nature Speakers Series event tomorrow night at 7 p.m. at Schnitzel's Restaurant. And this event will feature River Institute research scientist Matt Windle, who's going to be talking about some of our recent collaborative shoreline restoration work along the St. Lawrence River. And this includes a Phragmites management project at Rotary Creek. So if you're in Cornwall, please come on down to Schnitzel's Restaurant. And we also will be live streaming the event on Facebook. So I will also put that link in the chat if you're able to join. So with that, I'm really happy to introduce Meghan Ward, who is a PhD candidate specializing in wetland conservation at Trent University, where she studies the effects of invasive Phragmites removal on Great Lakes coastal wetland species. She'll be sharing some of her PhD work, which looks at changes in plant and insect communities after Phragmites removal. And just a few technical or logistical notes. Questions are really welcome, so please feel free to type any that come to mind in the chat, and we'll come to them at the end, and we'll leave plenty of time for questions. And also just a note that this webinar is being recorded and will be posted on the River Institute YouTube page. So yeah, I'll hand it over to you, Megan.
Okay, hello everyone. Thanks for coming and listening to me talk for the next 55 minutes or so. I'm going to share a little bit about my PhD research, like what Elsie just sort of mentioned. I study the effects of Phragmites removal on Great Lake coastal wetlands. So all of what I present to you guys today is going to be data that was collected from Point Pelee National Park. I study Phragmites removal at Point Pelee National Park. I also study Phragmites and the invasive hybrid cattail type of glauca at Point Pelee National Park and Lynn Shore's conservation area in Whitby. So those are my two sites, but today I'm going to talk to you about results we found specifically from Point Pelee. So Point Pelee, if anyone has been there, is an absolutely beautiful national park. It's actually Canada's smallest national park. And it's just on the most southern point of mainland Canada, along Lake Erie. It's one of the largest remaining Great Lake coastal wetland complexes that we actually have still in Canada, and an absolute privilege to spend my summers walking around and wading around these wetlands. It's a wetland of international significance. It has a Ramsar designation, which is an international designation. But despite how important and internationally recognized these wetlands are, they've been invaded by two primary invasive species, mostly this Typha glauca, this hybrid invasive cattail, and Phragmites, which will be the point of the talk today. And so the figure on the left, this sort of map, shows an image from some research that was published a little while ago, but how we've seen the infilling of a ton of these small open water ponds. And these open water ponds are critical habitat to fish and birds and reptiles and amphibians in wetlands. And Phragmites and Typha combined fill in these small ponds and these small areas of open water. And so when you look at figures like this over time from 1959 down to 2010, you can see that the marsh has completely changed. And so in response to these changes that they've seen at Point Pelee National Park, the staff there have done a ton of restoration work. And it's this restoration work that I'm going to present on. And so in the north section of the march, they've been removing Phragmites manually using the cut to drown method, where they cut Phragmites below the water level line to allow the rhizomes to drown out. They removed all the biomass by hand and then burned it off-site. They've also started creating channels where pre-existing channels used to exist before they were all encroached upon as well. And I'll talk about some of these different restoration works that have been going on. So the first sort of project I want to talk to you about is in the southern section of the marsh at Point Pelee National Park. So this is where Point Pelee staff removed just shy of 10,000 meters squared of Phragmites in 2020, so exactly 5 years ago. They went in and, as I said, used the cut to drown method and took out all that dead remaining biomass and burned it offshore. So no biomass, no dead material or dead stems or dead leaves was actually left in the wetland. It was all removed. And I think that made quite a significant difference for some of the results that I'll share with you. Now, this was an academic's, perfect location because now we had three different types of sites. We had a Phragmites invaded site where Phragmites was still present and had never been treated. And that was just north in this area. And then we had a Phragmites treated site where Phragmites still, where no Phragmites existed anymore. We used to have a patch of invasive Phragmites, and that is where the team removed about 10,000 meters squared. And then south of both of those sites, we had a non-invaded site. We had a reference original buttonbush thicket swamp that for some reason, still actually unknown to us, Phragmites just never invaded. And so because we had these three sites, we could ask some really interesting questions. We could compare plant and insect communities in really interesting ways. And this is the first goal or project that I want to present to you guys today. So our goal was to explore changes in vegetation as well as emerging aquatic invertebrate communities after the manual removal of Phragmites. So we collected vegetation surveys at all three different wetland types. We used 1 meter square quadrats that are pretty consistent and available across literature and science for sampling plants. And we identified all the different plants that were present, as well as any remaining Phragmites stem density in these non-treated wetlands. And I was also really interested in looking at the insect community because while plants are, of course, very important, especially when you're looking at the restoration of Phragmites, so too are the insect communities. Learning about insect communities can tell you about food webs and trophic dynamics, how energy is moving through different animals in the wetlands, and it's quite critical. And so we wanted to look at emerging aquatic invertebrates. And so those are invertebrates, usually insects, that have a larval aquatic stage and then often an aerial adult stage. So think of something like a mayfly or a dragonfly or a damselfly. They have larvae that live in the water that then climb up stems of plants and emerge with wings and then fly around. And so when those larvae are climbing up the stems of the plants, we use these traps that sit right on top of the water's edge, and we can collect all those insects that are flying around. And then we can sample different insect communities across these different sites. And then once we had our insect communities already sampled from the wetland, we took them to the lab and identified them. We could collect abundance, so just how many individual insects there were, as well as more community-based data, how many of different species or different families were present. So we did our best to identify everything in the lab as we could. It turns out identifying insects is incredibly difficult, especially when you're a novice like myself and you didn't have any experience. So we partnered with Nature Metrics to do DNA identification of these insects as well. So we had both lab-based identification and DNA identification. And so at the end of this summer, this was all data collected in 2022, so 2 years after restoration, we had some really interesting results. We had data from the Phragmites invaded site, we had data from the treated site, and we had plant and insect data from this non-invaded reference site. So it made a really interesting site for comparison among these different areas. We could answer questions in terms of, what happens to the plants and insects when Phragmites is removed, and do we eventually see a return to native communities when these animals or when these invasive plants are removed? And these are the results that I want to share with you today. So in terms of vegetation richness, which is just the number of different plant species that we saw when we were sampling, as well as abundance, how much of those plant species were present, these were significantly highest in the non-invaded sites and the treated sites, which is a great finding only 2 years after treatment. And we found that plant richness, so the number of individual plants again, was way lower in these. Pretty much all we found was European frogbit, which is another invasive species, and duckweed. Here and there, a few other plant species, but otherwise really not much. And you can just see from this photo, these are photos of 3 different sites that we sampled. It's pretty clear where the plant diversity drops off. Invertebrate richness, so the number of different insect families per meter squared per trap that we were able to sample, was way higher in these non-invaded sites and these treated sites. And we saw that number drop quite significantly in the Phragmites invaded site. So not only are we seeing plant richness decline, but we're also seeing insect richness decline. So way less different types of insects when Phragmites has invaded. Invertebrate abundance, which is just the number of insects per meter squared, was really high in this reference non-invaded site, followed then by the treated site, and way lower in the invaded site. And this is important because a lot of insect abundance feeds things like marsh birds and marsh frogs and turtles and snakes. And if we don't have the abundance of insects available, those other species higher up the food web or food chain can die because they just don't have enough prey available. So this finding is really important. It shows us that if we do take out Phragmites, those insects come back, those bugs come back, which is critical to good restoration and positive long-term goals. Now, I'm going to share some stats in case anyone in the room is a stats nerd, but don't freak out. If you don't like statistics, like most people, you can just look at the figure and I'm going to walk you through it. So we have Permanova analysis of similarity results on the right there if you're interested in looking at those variables. All that tells us is that our data is significant. So the trends that I'll describe are statistically significant. So I want you to focus on the figure on the left. This is a NMDS, which is an approach to do community composition analysis. So how do the whole communities of the vegetation change among these different sites? Are there the same species that are present? Is the amount of those species present among the different sites? And how does that change? How much variability is there? Variability in species richness is all really important to functioning, healthy ecosystems. And so what this figure on the left shows us is that there's a lot of overlap between blue and green, which means the vegetation communities are really similar between the treated site and the non-invaded site, which is a fantastic result 2 years after treatment. This means that previously this treated site was full of Phragmites. Now native species have started coming back, and it is really similar to this non-invaded reference buttonbush thicket swamp. So this is a really positive finding only 2 years after treatment. You'll notice that the pink colors are all quite tightly clustered. That circle surrounding them, it's called an ellipses. It's not very big. It means that there's not a lot of variation in the plant communities in these invaded sites, and all these sites look really similar. They're pretty much all characterized by lots of Phragmites, lots of dead Phragmites material, and H. rennae, which is the invasive frog bit that I mentioned before. And so this result is really positive. 2 years after manual Phragmites removal, we saw the plant communities returning to what they would have been pre-invasion, or at the very least, what the other reference buttonbush thicket swamp looked like. So this is a positive story, a positive outcome for Phragmites management. And when we look at the insect community composition, we see different trends, but these trends are actually still quite positive. So we see there's quite a lot of distance between the blue and the pink, which means the insect communities in the treated site are now very different from the insect communities in the invaded site, which makes sense. Phragmites provides very different structure in its ecology and in the habitats to something that is full of native plants in a wetland. And again, there's just not a lot of different types of insects in these Phragmites patches. And so the general takeaway we have from this figure, from this story that this analysis tells, is that insects are just a little bit delayed behind the plants in returning to pre-invasion status, which makes sense. When we look at restoration ecology and successional ecology, we often see plants responding first and then insects following a couple of years later. And so this figure kind of tells that story. But it's interesting because when you look at the ecology of these different insects that are in these habitats, it actually makes sense. So Scardidae is a marsh moth, and it breeds and forages and raises young in areas full of what's called detritus, dead plant material or decomposing plant material. And so it makes sense that you see this insect in areas that are heavily invaded by Phragmites because Phragmites produces so much dead material and so much biomass every year. However, when you look at something like Distictidae, this is a predaceous diving beetle. Predaceous diving beetles lay their eggs for their young on the bottom of floating plant leaves. So think of something like a lily pad that rests just on the surface of the water. It's underneath that pad that the Distictidae are going to lay their eggs for future generations. And that habitat structure just doesn't exist when Phragmites has invaded a site. There is no more open water area. There's no more water coverage. What we often see is just hard, thin stems of Phragmites coming in and out of the water. So when you look at the ecology of the invertebrates, it also makes sense when we think about how Phragmites changes and negatively affects different wetland compositions and different habitat types. And so this was our first goal, and I'm going to provide some of the key summary takeaways. Phragmites can be successfully manually removed from Great Lake coastal wetlands at small scale. So 10,000 meters squared sounds like a lot. Compared to other restoration projects, it's actually a relatively small site, but we saw really successful manual removal of Phragmites. The vegetation community of the treated wetland reflects the vegetation community of the non-invaded reference wetland, which is the ultimate goal of restoration ecology. If we can have the site that was restored be really similar to a site that is still healthy, that's an awesome takeaway for restoration ecology and for Phragmites management. And that's what we found here today as well. We also found that the insect communities of each wetland are pretty unique on their own. The invaded wetland supports the lowest abundance. So this is just more data and more research that shows Phragmites is negatively affecting not just birds and other plants, but also insect communities. And the abundance of vegetation and invertebrates were highest in the treated and non-invaded wetlands. Again, a really good pat on the back to the work done at Pelee because this is a really successful restoration project. Now, we're talking about invasive species, so it can't all be too positive. I am going to chat a little bit about our second goal as well. Our second goal also took place at Point Pelee National Park, but this time in the northern area of these wetlands. And we wanted to assess the method of rolling Phragmites in non-inundated habitat. And so our question was, how does the treatment method of rolling Phragmites stems affect both Phragmites grow back, so next year Phragmites coming back, as well as native plants in that area? Rolling is a method that is in the best management practices for Phragmites, but is actually recommended to use in addition to something else, perhaps in addition to burning or mowing or often herbicide use. And oftentimes, rolling is done in aquatic systems where there's lots of standing water. So rolling just means cutting the Phragmites, often at ankle height or so, pushing it over, often into the water, and allowing all the microbes and the bacteria in the water to break down the Phragmites. And it can decay within a year or so. And this works really well in inundated habitat or habitat that has standing water. However, at Point Pelee National Park, I had mentioned Typhloglauca was a big problem, this hybrid invasive cattail. This cattail has created floating mats of dense vegetation upon which Phragmites is growing. And so these areas aren't actually saturated with water. There isn't actually any standing water in these habitats. And so when these sites were rolled with no other treatment method applied, the Phragmites stems wasn't saturated. There wasn't any aquatic microbiome or bacteria or any little insects and bugs that could come and chew up the plant. They were just left in dry areas of thatch. And so this provided the very first instance of ever assessing rolling as a sole method of treatment in dry sites, in sites that don't have any water. And so this is where that work took place. We had 3 sites that were rolled, 3 patches of Phragmites on this floating type of mat that was above the water, where Phragmites was rolled down. And that's in the blue on this map. In the red on the map is pre-existing Phragmites patches that have not been treated. And so we were able to compare areas with Phragmites to areas that had been treated for Phragmites. And we compared things like Phragmites stem density and native vegetation abundance and richness, so the different types of species that were present. And so just to give you a visual of what some of this looked like, because I think that will make quite a big difference. As I'm sure many of you are familiar with being in a frag patch or walking or driving by Phragmites patches, this is what they often look like from the inside. So this is July of 2022 in these invaded sites before being treated with rolling. You can see there are tons and tons, often more than 100 stems per meter squared of Phragmites, both live and dead, taking up a lot of space. And one of my poor summer students trying to get insect containers in these sites as well to collect some bugs. And this is what these sites looked like after rolling. So in May of 2023, this was the first year after rolling, there wasn't much growing back because there's not much growth in May at Point Pelee. But in July in Point Pelee in 2023, we can see vegetation did start to grow back. And unfortunately, almost everything you're seeing here is more Phragmites. So these are the treated sites that just have more Phragmites growing back. So not visually, at the very least, looking too positive. You may also notice this dense amount of thatch. If you can see my cursor, this dense amount of thatch in the middle of the photo in July, that measures almost over a meter thick. So over a meter of dead Phragmites material is just left to decompose without any water in a dry site. And I have a few more photos here of Phragmites. You can see how thick these patches are, how some of our meter sticks are over 60 centimeters, and how their only species growing back again in July is unfortunately more Phragmites. I'm going to try and play this video for you as well of one of our summer students walking through this area, and you can see she's just kind of bouncing on this thick layer of dead material. Let me see if this video will work. So you can see how thick that is there for walking through these invaded sites. So quite a lot of dead material, unfortunately. So let's get into some results. We looked at native plant richness, so the number of different plant species after rolling. And we compared that to pink and blue. We compared that among these control sites where Phragmites had invaded and no treatment took place, to the treated sites in the blue where Phragmites was previously there and then it had been rolled as a treatment method. So we have data collected from 2022, 23, and 24. 2022 is before any treatment took place, before any rolling occurred. And we don't really see any significant difference in the number of different plant species among the treated and control sites, which makes sense because right now at this point, they're both fully invaded sites. When we look at 2023, we actually see less native plants, other than Phragmites, of course, less native plants in areas that have been treated versus in areas that were invaded by Phragmites and just left alone. So this first year of data was not promising find. Native plant richness actually declined in areas where treatment had taken place. And unfortunately, in 2024, that trend continued and increased. We saw even less native plants in 2024 in areas that had been treated versus in control areas where Phragmites was just left to grow. So this is really not promising find. And what it tells us is that likely all this dense thatch, all this dead, thick material that had been collected was too thick, reduced too much light penetration, changed the soil availability and water availability, and native plants just could not penetrate through it, and native plants could not survive. But we did see lots of Phragmites in the photos that I showed you. So when we compared Phragmites' live stem density, so this is only live plants, not any dead plants that might have been standing, in 2022, before any treatment took place, there wasn't any significant difference among these two sites, the control sites and the treated sites. And unfortunately, in 2023, so this would be the year after treatment took place, we saw more live plants growing back in the treated site. So there were less live plants in the treated site compared to the control in 2023. But the best finding, if this restoration work had been successful, would be that there would be no blue in 2023, right? No live Phragmites stems coming back in 2023. And unfortunately, that's not what we found. Essentially, Phragmites was the only plant able to grow through its own thick, dense thatch accumulation. And unfortunately, in 2024, we saw even more live plants coming back. And what we can infer from this figure is that over time, these areas that were treated for Phragmites are actually going to have the same amount of Phragmites back as these control sites that had been never treated for Phragmites. And so these areas that had the rolling are eventually going to look like areas that had never even been treated in the past. I anticipate that change occurring even within 3 or 4 years after treatment. So 2025 and 2026, we're going to see the return back to what this would have been like before. So again, this is not necessarily a positive finding. However, it teaches us a lot about best management practices for Phragmites, specifically in dry sites. Now, when we look at total Phragmites stem density, this is dead stems that remain standing, as Phragmites often do for a few years, as well as live stem density begins. We can look at those changes over a couple of years as well. So in 2022, there was no difference in the number of live and dead stems in the control sites versus our treated rolled sites. In 2023, we see the comparison become way smaller, way less number of total Phragmites in 2023. And that's because in 2023, in these treated sites, it's only live plants. Everything else was pushed over. So any dead plants from previous years that had been standing have been rolled. So this total Phragmites stem density in 2023 truly only represents live stem density because that's all that's still standing. Unfortunately, though, in 2024, these values increase again. And so this is telling us that even though there is a lot of dense thatch accumulation and layers of dead stems, plants that had grew in 2023 were still standing in 2024. And so this idea of biomass accumulating, dead plant and dead stem and leaf accumulating over time was still occurring even in these treated sites, which is again, not beneficial in terms of the ecology of the site and the actual plants and insects that live there, but hugely informative for best management practices and for these types of webinars about teaching folks from our own mistakes. And so the second goal was to again assess rolling as a potential method in dry sites. And here's what we found. Rolling is not an appropriate method of Phragmites removal in non-inundated sites, so dry sites. Lots of research has been done about rolling in aquatic sites, and it works. The invasive Phragmites biomass and dead material decays over the course of a year, maybe 2, and that's an okay method for treatment, but not in these dry sites or these terrestrialized sites. Unrolling in non-inundated sites causes a sustained decline in native plant richness. This part is key. We didn't just see native plants decline one year and then rocket back up, but we saw them consistently decline in abundance over time and in diversity over time, which is not a trend that we want to see happen across different wetlands and different treatment areas in Ontario. And unfortunately, Phragmites continued to persist in 2023 and 2024 after treatment occurred in 2022. So this whole second goal of the summary is all to say it is likely not a good option to roll Phragmites as a sole method of treatment in dry sites or in sites without any standing water. Additional treatment methods definitely are required. And so that wraps up my presentation to you guys today. I really appreciate you listening to some of the research that I found over the past few years. If you have any questions, of course, you can ask them now, and I'm happy to chat about you or chat with you about them. But if you do want to email me or get in contact with this research, you can email me there. The literature from this research, so the scientific papers that we're producing from this work, have all been published, aside from one paper that is just pending review. And I'd be happy to send that around if anyone wants more of the technical and statistical approaches to some of this work. But thank you very much for listening.
Well, thanks so much, Megan. Really appreciate the presentation. So I'm happy to open up the floor for questions. Feel free to either type your question in the chat, or you can also, I believe, raise your hand and we can unmute you to ask your question directly. Thanks, Bill. I guess in the meantime, I do. Oh wait, here.
There's some questions.
Okay. I think we've got Aaron, so maybe I'll. There, Aaron. I think you should be able to ask your question live now.
Yes, I can. Thank you. Thanks, Megan. That was really good. Really interesting to kind of see the experimental removals you've done. I just had a question about the first one with the timing. I might have missed it, but you said the removal happened in 2020. What year or years did you do the vegetation studies, like follow-up vegetation studies and invert studies?
Yeah, so they were all collected 2 years after treatment. So in 2022, we went in and collected that data. That was actually data for my master's, and my PhD has since collected 4 more years of data. So we're currently working on analysis over like a 5-year since treatment data set. But what I shared today was just 2 years, the single year of 2022 after treatment.
Okay, thanks. Yeah, because that'll be really interesting to see the long-term effects. So
yeah,
it's exciting to hear that you're looking into that.
So we have a big question from Jackie, and Megan, I'll let you decide how to answer this, but Jackie asks, do we know what the best method is to eliminate Phragmites?
That is quite a big question. I would say we don't necessarily know the best method because there is no one method that's perfect for every site. A big part of sort of best management practices and a lot of research coming out with Phragmites now is saying that, a lot of this treatment we have to do is site specific. So whether there's open water or there isn't open water, whether more Phragmites surrounds the corridor of where you're treating or there's no more Phragmites present, whether or not you have a robust native seed bank that's ready to come in and grow after the Phragmites has been removed, or maybe there is no more native seed bank and so reseeding is needed. There's no one catch-all treatment method for Phragmites because it depends, unfortunately, on the site, depends how long the Phragmites has been there, and it depends on ultimately as well the finances and the time of the management. So I'm sure that's not the answer we're all hoping for, but I think that's the closest to truth that we can sort of get, at least with what we know right now about management and restoration.
Thanks, Megan. I realized I missed a question from Tyler. Have you evaluated areas treated with herbicide applied by UAS. I think that might be drones. But Tyler,
So
feel free to raise your hand if you want to hop on. But go ahead, Megan.
So I have not done any research with herbicide treated sites. I'm sort of specializing a bit more in manual and mechanical removal, but there is a lot of research coming out of Long Point, looking at herbicide application, and actually a recent publication that I believe is open access, which means anyone can access it without having to pay for it, using drones to apply herbicide. A recent paper came out last year, maybe, but I don't do any work with herbicide, mostly because when Point Pelee was applying for all their funding to do restoration work, herbicides actually weren't approved in Canadian national parks at the time. So they had to resort to non-chemical means, and that's where my data collection took place. So therefore, I studied non-chemical applications.
Great, thanks. So a question from Anna. In the manual removal, is there evidence of regrowth, and how often do you expect to have to return to a site to maintain its integrity?
Yeah, so there is regrowth of Phragmites in the manual removal. So specifically talking about the first goal where they removed Phragmites and we could sort of measure and compare stem density and Phragmites in a treated, invaded, and non-invaded site. In the treated site where they removed 10,000 meters squared, they did have Phragmites grow back just like one plant at a time. And so for 3 years following treatment, like their big treatment effort in 2020, they had a team of volunteers come in and just manually cut individual stems, and it was less than 10 to 15 stems per year. But it is expected that with any type of management of Phragmites, some type of long-term commitment is needed, but the assumption and what has sort of already been proven is that the initial cost that has to be spent reduces over time. That's sort of that goal, that we're not consistently spending as much time and effort and money as we do at the initial outset. But they did still have to do a little bit of like individual stem management.
Yeah. Great. So a question from Fred. Have the biocontrol moths been introduced at Point Pelee?
They're going this summer. Yes,
Oh.
so Pelee is working with I believe it's the team at the University of Toronto with Dr. Ian Jones. And they are planning to release the biocontrol moths definitely in the north section. I shouldn't say definitely because I'm not a Point Pelee staff, but from what I've heard, very likely in the north section of the marsh. That's actually where the most heavily invaded area is, in the northern area there. Point Pelee is kind of like a long triangle. So at the larger bit at the top, that's where a lot of Phragmites is. And they are hoping to pepper the moths in a few other patches as well.
Great. And while you're answering the next question, I might try to find a presentation on the biocontrol moths to put a link to in the chat, just in case other people aren't aware of that program. So just a question from Sukrit. So your presentation mentioned the rolling method. What is the best time in the year to use this management practice? And so maybe you want to think about using it in aquatic or inundated sites, in addition to the terrestrial one, because it sounds like the terrestrial one wasn't the generally recommended.
Yes.
Yeah.
If you have a site that has like lots of open water and you're confident that there's bacterial communities that can help decompose the invasive Phragmites, which there likely is if you're in a wetland or a lake and you have open water, then there's a few things you need to be sort of aware of. What you don't want to be doing is disturbing Phragmites where the seed propagules, that fluffy bit on the top, are still ready and healthy to reproduce, because that will help spread Phragmites. So the best option is to either do it at the end of the season once all of that's done, and thinking like September, October or so. So fall treatment is when the staff at Point Pelee did the rolling. It's when a number of other folks with Ontario Phragmites working groups do their methods or their treatment methods. And that seems to be working okay. But one thing to sort of keep in mind with rolling is that if you're not combining it with something like herbicide, by the fall, the rhizomes, so those really intense root networks that are underground, often we can't see, have now started storing all their carbohydrates and their sugar. So all their energy is now being stored in the still living root network. And so just cutting the dead stems will allow you to take biomass out, especially if you're removing all that dead material, but it is not necessarily solving the root problem of where the energy is, and that's why we see so much return following year. However, herbicide application followed by rolling a little bit later, that could be okay. That could be a method where, the herbicide is sprayed onto the leaves take in the herbicide gets pulled down with the carbohydrates and with the sugars into the roots, and it kills all the root networks. And so that can work. But rolling on its own, especially in non-inundated sites, I would recommend even once the seed heads have expired.
Thanks. And I saw Sukrit give you a thumbs up there. So a question from Shauna. I've heard about using rolling cutting before more frequently, for example, 3 to 4 times per year for multiple years in attempts to exhaust the plant's energy. Is this something that you've looked at?
It's not something that I have studied myself. I have seen some papers about like mowing or cutting multiple times a year to do what you're sort of describing here, Shauna, and that you continuously remove sugars and carbohydrates from the biomass so the plant doesn't have as much storage over time. And that can work. However, rolling alone or cutting alone often encourages regrowth of plants. And so even in native plants, cutting is often a method to get more growth later on for the next generation. And so I would be cautious with consistently cutting Phragmites over time with no other management application. Perhaps if you're burning or if you're applying herbicide, then that might be a different situation. But consistent cutting can actually encourage more growth, especially in the next season. So just be cautious with that if you're not combining it with any other treatment method.
Great. So if anyone has any additional questions, oh, here we go, from Fred. Are there any native Phragmites at Point Pelee?
Yes, there are. So there's at least 2, if not 3 or 4, like confirmed native patches of Phragmites in the park. I was actually chatting with someone yesterday about native Phragmites and how it does seem to be popping up across the province. And there's actually been a few instances of folks submitting biological samples that they assume are from invasive populations that end up actually being confirmed native. And so we do have native Phragmites at the park. One of the reasons why the park is trying to move forward with a lot of this restoration work is so that those native populations aren't encroached upon and aren't outcompeted. But then, of course, because we have the native and ton of the invasive, I think there is a bit of worry now about a hybrid potentially popping up at Point Pelee as well, as there has been in Las Vegas and Minnesota and a few other locations in Ontario too.
Yeah, and for anyone who's not familiar, at least in previous years, the Invasive Species Center has offered a program to do genetic testing for Phragmites. If you have a patch that you want to confirm whether it's invasive or native, and keep an eye on their website if they're going to run it again this year. So yeah, if anyone has any additional questions, again, please feel free to raise your hand and I can unmute you. I guess I have a question. Is Point Pelee, have they continued or expanded the Phragmites removal since your research concluded?
So they haven't done too much more Phragmites work. Part of the issue with Point Pelee is that there is just so much Phragmites now. Aside from herbicide application, manual removal is it just seems silly at this point, to be honest, because there's so much dense Phragmites. In the north, there's just kilometers and kilometers of patches that are incredibly dense, which is why they've moved a bit more towards the biocontrol project, because that seems to have a bit more reasonable long-term management goals, and that's sort of where Pelee, I think, is heading into these ideas of, how do we manage this long term? What Pelee is doing a lot more now, though, is focusing on the type of glauca. So this hybrid invasive cattail that forms these floating mats. And these floating mats are like 3 meters thick, if not 4 meters thick of detritus and root material. We can walk on them and it feels like you're walking on the road. Like you're not really even buoyant. There's no holes in it. And this invasive typha combined with the invasive Phragmites has really changed the marsh. That's where all that open water has been lost to and they've all encroached in. And so Pelee has put a lot of effort right now into cutting channels into these ponds into these floating mats where there used to be natural open space. So that's what a lot of their work for the last summer, previous summer, and then this upcoming summer has been in dealing with these floating typha mats, because it also seems that floating typha mats are just perfect habitat for more Phragmites. So it's kind of, two birds, one stone if you can stop the encroachment and reduce some of that habitat suitability for the invasive plants.
Great.
Yeah.
And I guess just a quick follow-up question then, and based on your research, is there like a sort of maximum area of kind of dense Phragmites stands where you'd say that manual removal is like a really feasible option, or?
Yeah, so 10,000 meters squared. The folks at Pelee did that with about a team of 5 over the course of a few weeks, and that has worked out really well for manual removal. And I know there are some teams, like Janice Gilbert leads a lot of Phragmites removal in Ontario, and she'll work consistently throughout the summer and into the fall, treating larger patches that are manual. Some folks aren't as interested in using chemical application for treatment, and so there's just this more internal assumption that maybe it's going to take a couple years of management, or maybe like a couple teams and a bit more funding. But it does seem to work. There's actually research that's come out that shows restoration at small scales makes really positive, significant difference. And so even treating these smaller patches of Phragmites, when we combine and look at, how Ontario is doing that together across all these communities, is making a lot of really positive progress.
Great. Well, I think that might be a really nice note to close out on. Oh, okay. One more question from Brian. When you cut channels in the cattails, does that increase opportunities for more Phragmites invasion? And I think Brian is referring maybe to some local areas where this has occurred. So do you have any experience from that?
Yeah, so invasive species in general, Phragmites and the invasive cattail specifically, do really well in areas that have been disturbed. And that's because there's often left behind open mudflat areas where no other seeds exist or no other plants exist yet. And because Phragmites and the cattails can spread so easily, it's their seed propagules that get deposited in these newly disturbed habitats. And so while we have seen Phragmites pop up in some of these areas that have been treated, we've actually mostly seen native plants return. That's a whole different chapter of my dissertation that I won't get into right now, but we have seen native species, native plants coming in and colonizing these areas. And because the Phragmites isn't as dense here, these other plants are able to come in, build root networks, store light, like they're able to access the light penetration and store carbohydrates and nutrients. So it has been beneficial the past 4 years of data that I've collected there.
Great. Well, maybe we can have you back some other time to talk about
Next
that
year.
part of your dissertation.
Yeah.
Okay. Are there any other questions? Okay. Well, if you do have questions, or again, if you are interested in becoming involved in some of the Phragmites PMAs, particularly in Ontario, please feel free to reach out to me. I did add my email to the chat. And if you have any questions, I'm happy to pass them on to Meghan in the coming weeks. So, yep. Once again, thanks everyone for joining us, and please come back next month on February 11th for our presentation with Victoria. Bye, everyone.